Hi everyone, this is Bhagilakshmi, Department of Biotechnology, KSRCT. So in this video, we are going to discuss about biofertilizers, their concept and need nowadays, and their role in improvement of crops, and what are the microbial organisms they are used as a biofertilizers for crop improvement, and mass cultivation process and commercialization. What are the carrier materials have been used in biofertilizer production, and their application methodology and advantages and disadvantages we are going to discuss in this video. In the last century, chemical fertilizers have been widely used in agriculture in tons. So the farmers were very happy at that period because of they got increased yield in agriculture in the beginning. But later, very slowly, the chemical fertilizers started displaying their own ill effects, such as leaching out, polluting water basins nearby, destroying microorganisms, beneficial microorganisms, friendly insects, making the crop more susceptible to the attack of diseases, reduces the soil fertility and thus causing impermeable or irreversible damage to all over the system. Then everybody nowadays they are talking about biofertilizers, organic manuring, organic products. So biofertilizer is nothing but it's a substance which contains living microbial organisms. When it is applied to seed or plant surface or soil, it will colonize the rhizosphere portion or the interior of the plant, and it will promote the growth of plant by increasing and supplying the necessary nutrients to the host plant. They add nutrients through the natural process of nitrogen fixation, solubilizing phosphorus, potassium, and also it stimulates plant growth through the synthesis of some important growth promoting substances. They are the natural fertilizing organisms, nothing but they are microbial inoculant of bacteria, algae, fungi, separately or individually or in combination of any two or three microbial organisms. This will help biological nitrogen fixation and give nitrogen requirement to the plant, and so that crop will grow and give yield. They help in building up of soil microbial organisms, thereby the soil health will be improved. It also includes organic fertilizers such as organic manure. Uses of biofertilizers can be recommended for improving the soil fertility in organic farming. What is the need and concept for this biofertilizers? It will improve the physical chemical nature of the soil, because in this biofertilizers there is no chemical will be added. This will consist of only a group of inoculant microbial organisms, bacteria, fungi, algae, like that. So they colonize more. It will secrete growth promoting substances also. It will attract more microbial organisms near to that area. By that way, it improves the soil fertility and the soil nutrient ratio have been maintained. And also, it will add organic matter to the soil. It will solubilize insoluble nutrient materials such as phosphate solubilizing bacteria, potassium solubilizing bacteria. When we add, the sources will be in insoluble nature in soil. Crops cannot directly absorb the insoluble form of these potassium and phosphorus. Potassium solubilizing bacteria, phosphorus solubilizing bacteria, by secreting various enzymes and organic acid, these non solubilizing sources will be converted into solubilized form so that plant can easily take up and it grows. It increases the availability and utilization of nutrients for the plant. It also secretes plant growth hormones, which is necessary for the crop improvement. It induces the disease resistance in crops. It germinates. Germination percentage of seed increases, inducing flowering, fruiting of crop. Reduced input cost and also increases the crop production. So these are the concept of why we have to add biofertilizers. These are the types of biofertilizers which includes nitrogen fixing bacteria, phosphorus solubilizing bacteria, phosphorus mobilizing biofertilizers, plant growth promoting rhizobacteria, and also biofertilizers for micronutrients. So in this picture you can see a different group of organisms: Rhizobium, Astrobacter, Azospirillum, PSM, and selective media plates, and their growth and zones also you can clearly visible around the culture. Coming to the type of biofertilizers, and the bacteria you have, it has been grouped as symbiotic nitrogen fixers, free living nitrogen fixers. In symbiotic nitrogen fixers, Rhizobium azospirillum species are coming, and free living nitrogen fixers we have Astrobacter and Klebsiella. Coming to algal biofertilizers, you have blue-green algae associated with the azolla, Anabina, Nostoc, Acetoria. In phosphate solubilizing bacteria, we have Pseudomonas, Bacillus megaterium. Fungal biofertilizer example, VAM, vesicular or vesicular fungi, vesicular or vesicular mycorrhizae, and also earthworms. Coming to the bacterial biofertilizers, the live cells, whole cells of bacteria can be used as a biofertilizer. So these microorganisms contain a special group of gene called NIF genes that is responsible for the nitrogen fixation. Atmospheric nitrogen has been fixed with the help of this NIF gene into the soil. So this will work under two conditions: symbiotically and non-symbiotically free living microorganisms. The symbiotic bacteria, this will have association with the crop plants, especially with the root nodules. The free living bacteria will be there in soil. It will not have any kind of association, but it live freely and fix atmospheric nitrogen to the soil. Coming to the symbiotic nitrogen fixes, example, the most important examples are Rhizobium azospirillum. Rhizobium, which lives in the root hairs of leguminous plants and forming a nodule. Plant root will give all essential nutrient to the bacteria it grows, and it synthesizes a new kind of substances. The name Rhizobium was coined by Frank in 1889. More than 20 cross-inoculation groups have been identified in Rhizobium called Bradyrhizobium. This is another one group, fast-growing rhizobia called Rhizobium. And this Bradyrhizobium is grouped as a slow-growing rhizobia. This is gram-negative, rod-shaped bacteria. Coming to the Azospirillum, another one symbiotic associated bacteria. You can see this Azospirillum in cereal plants. It inhabit both root cells and as well as surrounding of roots, and this forms a symbiotic relationship and increase the nitrogen fixing potential of the cereal crops. And this is recognized as a dominant soil microbial organisms. Nitrogen in the form range of 20 to 40 kg per hectare in the rhizosphere in non-leguminous plants such as cereals, millets, oilseeds, cotton, etc. So by using this azospirillum, considerable quantity of nitrogen fertilizers of 25 to 30% can be reduced or saved by using this azospirillum inoculant in soil. And these microorganisms have been commercially produced for the use of nitrogen supplying biofertilizers. And coming to the free living nitrogen fixers, we have Acetobacter and Klebsiella. So there are number of free living non-symbiotic bacteria have been isolated and studied from the soil. It is not associated with any kind of fruit hairs, root of leguminous or cereal crops. As such, you can isolate directly from the soil. Example, Astrobacter and Klebsiella, which provides the required amount of nitrogen to the plant from the soil. So these organisms also take part in nitrogen fixation, atmospheric nitrogen fixation. Coming to the Azotobacter, it's a heterotrophic free-living nitrogen fixing bacteria. This is present in alkaline and neutral kind of soil. The most commonly occurring species in arable soil of India. Apart from its ability to fix atmospheric nitrogen in soil, it can also synthesize some of the growth substances, promoting substances such as auxin and gibberelling, and also some vitamins also has been produced by the Astrobacter group. Many strains of Astrobacter also produces some fungicidal substances against some genera of fungus. And this is highly worked with rice, maize, cotton, sugarcane, pearl millet, vegetable and some plantation crops. So the yield growth will be more and also yield will be more when we apply these kind of bacteria in soil. It improves seed germination and plant growth also. And it requires organic carbon more for their growth. Coming to the silicate solubilizing bacteria, so most of the silicate and potassium sources will be there in soil. It will be of unavailable form. So the microorganisms which are present in the soil in that particular area, by secreting enzymes and number of organic acids, they degrade, dissolve it, and make them available for the plant. Okay. Plant growth promoting rhizobacteria, we have another group. In this group, they colonize roots of rhizosphere soil and beneficial to crop by producing plant growth promoting substances. Coming to the mass production of biofertilizers, this is one of the crucial step in biofertilizer production. So the isolated bacterial cultures were subcultured in nutrient broth. The cultures were grown under shaking conditions of 30 plus or minus 2 degrees Celsius and will be incubated until it reaches maximum population of 10 to the power of 10 to 10 to the power of 11 CFU. So under optimum conditions, the population level will attain within 4 to 5 days for Rhizobium, 5 to 7 days for Azospirillum, 6 to 7 days for Astrobacter. Then the culture will be obtained from the flask. This is called as starter culture. For large scale production, the starter culture will be added into the fermenter or in large scale vessel. The required quantity will be added. So appropriate media for that particular bacterial growth will be prepared and the specific inoculant that is required quantity will be prepared. Inoculated with the specific bacteria strain under aseptic condition will be done. Incubated for 30 to plus or minus 30 plus or minus 2 degrees Celsius and maintained for 5 to 7 days in a rotary shaker. This is most important. And then we have to observe the growth of culture and we have to estimate the population. Then the above media is prepared in large quantity. So up to this, we have prepared only the starter culture. After preparing the starter culture, we have to transfer into large quantity, which is in the fermenter. So the medium will be prepared in large quantity, sterilized and cooled, and then inoculated with the log phase culture, which is present in the starter vessel. We have to take appropriate quantity Usually 1 to 2% inoculum will be sufficient for this large scale production that will be added into the fermenter. And the cells are grown in fermenter by providing proper aeration, continuous stirring. It will grow more population will be grown in that particular condition, optimum condition. In between we have to check the population of inoculated organisms. Then later finally the cells will be harvested when it attained into the power of 9 cells per ml. After that we have to select a suitable carrier material. As such, you cannot apply into the soil or for the plant. It has to be mixed with the carrier material for large scale application. So the carrier material has to be chosen wisely. So these are some carrier material have been widely used in biofertilizer: peat soil, lignite, vermiculture, charcoal, press mud, farmyard manure, and soil mixture are used as a carrier material. Among all these, neutralized peat soil, lignite are found to be a better carrier material for biofertilizer application. So, how ideal material should be there? Carrier material. It should be of cheaper in cost, easily available, and should have higher organic content, matter content. It should not contain any kind of toxic chemical. Water holding capacity should be of more than 550% and easy to process. So these are the points have to be noted while selecting carrier material. And after selecting a carrier material, that has to be neutralized and sterilized, and then it has to be spread in a clean dry metallic plastic sheet, and then bacterial culture will be which is taken from the fermenter will be added to the sterilized carrier material, and it has to be mixed properly by manually or mechanical mixture. And then after that the inoculants are packed in a polythene bag, then it is sealed with the electric sealer. And while packing, the bag should be consist of low density grade. Thickness should be of 50 to 75 micron, and that pack after packing the bag should have some kind of markings like name of the manufacturer, name of the product, strain number, crop to which recommended, method of inoculation, date of manufacture, batch number, date of expiry, price, full address, storage, instruction. Everything has to be mentioned, marked in the packet. Coming to the next group of biofertilizer is vesicular arbuscular mycorrhiza, VAM. This term mycorrhiza was first used by Frank. It is called as fungus root. This is having a mutualistic association between fungal mycelium and roots, plant roots, and it's a endotrophic in nature. It lives inside the cell, and the mycorrhiza formed by aseptate Phycomycetes fungi. This helps in the nutrient mobilization from outside, from the distance place to the roots. Example: phosphorus, zinc, and sulfur. This is having symbiotic association with the plant root and soil of fungus of the 7 type of mycorrhizae. This play a great role in inducing plant root. This swarm are symbiotic endophytic soil fungi which colonize the roots of approximately 80% plants. So this hyphae helps in retaining moisture around the root zone of plants. It increases the resistance to root-borne or soil-borne pathogens and nematodes. So in this picture you can clearly see, name itself you can understand, arbuscular mycorrhizae, vesicular arbuscular. So they have a vesicle and produces arbuscules. In this vesicle they store all kind of nutrients. Then with the help of this arbuscule, the nutrients will be supplied to the root cells, and this VAM mobilize different kind of nutrients such as copper, potassium, aluminum, phosphorus, manganese, iron, magnesium from the soil to the plants. So coming to the morphology, we discussed earlier that it has an external hyphae having arbuscules and vesicles. First, it penetrates into the root cortex. So this is the mechanism of action of VAM. This is associated with the plant root. First step is penetrates in the root cortex and spreads around the roots of the plant. The name indicates, I said earlier, that they have a sac-like structure called vesicles, which will store all nutrients like phosphorus and phospholipids. The other structure called arbuscule helps bringing the distant nutrient to the vesicles and root. How we can produce in mass? We have to collect BAM spores first. It will be mixed with the sterilized soil, and that soil will be filled in pots, and after filling it, the host plant will be transplanted in that pot. That can be kept for 3 to 4 months in greenhouse, and then soil in the pot along with the roots of host plant is macerated, dried till it attains 5% moisture content, and then dried soil inoculants used for field application. So this is how VAM can be propagated. So these are the uses of BAM. It will enhance the feeding area of plant roots as the hyphae spread around the roots. It mobilizes the nutrients from the distance to the root. It also stores with the help of vesicles nutrients with the help of vesicles. Also removes the toxic chemicals such as phenolics. It protects against number of fungal and nematode infection. It also increases growth rate of plants such as citrus, maize, wheat, etc. And also it reduces sensitivity of crop towards high level of salts and heavy metals. Coming to algae growth as a biofertilizer, this is the most important free-living nitrogen fixers as cyanobacteria, commonly called as blue-green algae (BGA). More than 100 species have been identified in nitrogen can able to fix nitrogen. So this takes place with a specialized kind of cells called heterocyst. This BGA you can see very commonly in rice field before planting. The rice field filled with the water, and you can see a green color growth called this blue-green algae. Unlike Astrobacter, blue-green algae are not inhibited by the presence of chemical fertilizer. There is no chemical fertilizer added here. Inoculation of just algae can result in 10 to 14% increase in crop yields. It's very easy to produce this kind of algae. Usually mass produced in cement tank without any kind of investment. A small cement tank filled with fresh water, and this will not require any kind of processing. Quite and cheap. Cost of 10 kg may be about rupees 30 to 40 only, very cheap only. But it is having higher beneficial against certain crops like vegetable, cotton, sugarcane. Some of the algal biofertilizers are Anabina, Nostoc, and Oscillatoria. In this picture, you can see the algae. Then next one is Azula as a biofertilizer. It's a tiny freshwater fern common in ponds, dishes, and rice field. So common waterlogging area, you can see green color water fern. This can be also used as a biofertilizer. For rice, majorly it is used in countries like India, Thailand, Korea, Philippines, Brazil, and West Africa. This azolla has been widely used as a biofertilizer for nitrogen fixing process. And also it has a relationship between fern, that is BGA, blue-green algae. In addition to the nitrogen, the decomposed azolla also provide potassium, phosphorus, zinc, and iron to the crop. So this biomass, azolla biomass, gets doubled within very shorter period, 5 to 7 days or by vegetative method, it can fix 40 to 80 kilogram nitrogen per hectare per year, and it acts as a good manure for flooded rice cultivation. It also increases crop yield up to 50 to 20%. This has been observed while fertilizing the rice with azolla. Hybrids of azolla also developed, and it grows faster, tolerant to heat and cold, and it picks 4 to 5% more nitrogen when compared with the normal wild azolla. Coming to the application method of biofertilizers, there are three important ways of using nitrogen fixing biofertilizer bacteria: seed treatment, root dipping, soil application. In seed treatment, the seed is the most common method adapted for all type of inoculant. It's effective, highly effective and very economical. And this has been practiced for Rhizobium, Acetobacter, Azospirillum along with PSM. This can be done with two or more bacteria in a combination also we can done. There is no side effect of this kind of seed treatment. Important thing is seed must be coated with the biofertilizers of Rhizobium or Acetobacter or Azospirillum. Even each seed is coated with a or layer of these kind of bacteria. Then the PSM inoculant has to be treated an outer layer of the seed. So this is a process of seed treatment. So this method will provide you the maximum number of population of each bacteria required for better results. So mixing of any two bacteria and the treatment of seed will not provide maximum number of bacteria of individuals. Root dipping, application of azospirillum with the paddy and vegetable plants. This method called the root dipping method is followed. This require the quantity of azospirillum will be taken and has to be mixed with a 5 to 10 liter of water at one corner of the field itself. We can prepare this, and all the plants can be taken and kept. This has been immersed in that water for minimum of half an hour before it sows. Before sowing process, it has to be mixed with the water. Then coming to the soil application, this PSM has to be used as a soil application. Use 2 kgs of PSM per acre has been finalized, recommended. This PSM with the 400 to 600 kilogram of cow dung, directly we cannot apply this into soil. It has to be mixed with the cow dung along with the half bag of rock phosphate if it is available. Rock phosphate, cow dung, and then PSM has to be mixed properly, and then it has to be kept in a shade or selling over for overnight and maintain 50% of the moisture. And then later you can take this mixture and can be added as a soil application in rows or during leveling of soil. You can add this kind of biofertilizer. These are the precautions you have to take care. The biofertilizer has to be stored in packets in cool and dry place away from direct sunlight and heat. This has to be taken care, and use a right combination of biofertilizers. If any kind of wrongly we use this biofertilizers, we won't get the benefit of this biofertilizers. Rhizobium is a crop specific, so use in a specified crop. So the recommended crop, recommended fertilizer has to be used. Do not mix with the chemical. It should not be mixed with the chemical. And use the packet before expiry itself only on the specified crop by the recommended method. So these are some precautions has to be followed while using biofertilizers. And these are the advantages of biofertilizers. We have already discussed. It fixes various nutrients like nitrogen, phosphorus, sulfur, and various micronutrients in soil. It maintains soil health and it's a pollution free. Secretes important hormone, growth regulators. It helps in higher crop production and it's a low cost only production, affordable price. Everyone can, every farmer can easily uh, uh, can get these biofertilizers, and it helps quick decomposition of organic matter. It reduces crop diseases due to the secretion of some antibiotics. It's easy to apply also. So these are the advantages of biofertilizers. Coming to the disadvantages, this requires special care while starting from the selection of strain, mass production, everything. It should not have any kind of contaminant organisms, and the special care has to be taken for long term storage, because it has to, it consists of only microbial inoculant, so that should be in alive condition, and so that only they have mentioned like it should be used before their expiry date. So if any contaminant organism is there in a carrier medium or any kind of growth, microbial growth, if any wrong organisms strains have been has used, so that will be a problem, and they are that much effect you cannot see in crop development. And this biofertilizers lost their effectiveness if the soil is too hot and dry. So soil also has to support. If a soil is very hot, dry, the biofertilizers will not be in effective condition. Coming to the conclusion of biofertilizers, biofertilizers helps in increasing crop productivity. It reduces the chemical fertilizers, it reduces these toxic, it maintains soil health, soil nutrients will be maintained, beneficial organisms load will be more, will be maintained in higher quantity in soil. It will replace 25 to 30% of the chemical fertilizers, and this can be applied very easily through seed coating, dipping, seedling inoculation, seed inoculation, then seed pelleting. By using carrier based, you can go for commercialization in large scale. Thank you.